Assalamu alaikum, dear students. Welcome to the course Principle in Animal Life 1. This is lecture number 57. The topic is the cell structures, mitochondria and peroxisomes. Discussing agenda for the topic will be mitochondria and chloroplasts change energy from one form to another. The evolutionary origins of mitochondria and chloroplasts. Mitochondria chemical energy conversion, and finally peroxisomes oxidation. Lecture outcome. After watching and listening this lecture, students will know about mitochondria and chloroplast, their evolutionary origin and functions, and furthermore they will discover the concepts related to peroxisomes. Introduction. Mitochondria and chloroplasts change energy from one form to another form. Organisms transform the energy they acquire from their surroundings. In eukaryotic cells, mitochondria and chloroplasts are the organelles that convert energy to forms that cells can use for work. Mitochondria, singular mitochondrion, are the sites of cellular respiration, the metabolic processes that uses oxygen to drive the generation of ATP by extracting energy from sugars, fats and other fuels. If you recall the lectures from 5th chapter of this course, we discussed aerobic cellular respiration in quite detail. Aerobic cellular respiration is a process in which the food is oxidized in the presence of oxygen and energy is released. So this process is completed in 3 stages. First, 6-carbon glucose molecule is broken down into 2 3-carbon pyruvate molecules, and then this pyruvate molecule converts into acetyl coenzyme A through link reaction, and then acetyl coenzyme A enters into Krebs cycle, and during this process ATPs are synthesized through substrate level phosphorylation, and the NADH and FADH2 molecules are also laden with electrons, and then these high energy electron carriers deliver their electrons to the electron transport chain, and in this process ATPs are synthesized through oxidative phosphorylation. So because oxygen is the final acceptor of electrons in electron transport chain, therefore this process is referred to as aerobic cellular respiration, and the main site where this process, electron transport chain and Krebs cycle, occurs is mitochondria. So therefore mitochondria is known as the powerhouse of the cell, and these are the substrates which can participate in this aerobic cellular respiration pathway. These molecules or their derivatives can enter at different stages into the aerobic cellular respiration process. Chloroplast, on the other hand, is only found in plants and algae, and they are the sites of photosynthesis. Photosynthesis is a process in which chloroplast converts solar energy to chemical energy by absorbing sunlight and using it to drive the synthesis of organic compounds such as sugars from carbon dioxide and water. So the greenish part of the plant where the chlorophyll is present. This process occurs. Carbon dioxide and water combines in the presence of sunlight, and glucose molecule is synthesized along with water, and plants are the primary producers, and then this glucose molecule is used by the plants for generating energy. This glucose molecule is then incorporated into aerobic cellular respiration in plants, and furthermore, when the animals feed upon plants, then this organic material is transferred to the animals, where it also helps in the production of energy. In addition to having related functions, both have related functions. They convert one form of energy into other form of energy, and they sort of generate energy. However, energy can not be generated, it can only be converted from one form to another form. So mitochondria, the energy which is liberated as the electrons move from one carrier to another carrier, is used to pump protons to the intermembrane space, and then as these protons move back to the inner lumen through ATP synthase, ATPs are generated. Similarly, the sunlight falls on the green part of the plant, and the process of photosynthesis occurs, where the sunlight energy is stored in the glucose molecule, in the bonds which are present in the glucose molecule. So this is one similarity between those two structures, that they helps in the conversion of one form of energy into other form. And the second similarity between the two is that they have similar evolutionary origin. Mitochondria and chloroplasts display similarities with bacteria that led to the endosymbiont theory. Endosymbiont theory was presented by Lynn Margulies back in 1967, and this theory explains the evolution of a eukaryotic cell from prokaryotic ancestors, and till date this is the most well accepted theory about the evolution of a eukaryotic cell. This theory states that an early ancestor of eukaryotic cells, a host cell, engulfed an oxygen using non-photosynthetic prokaryotic cell, and thereafter it established a beneficial relationship with this non-photosynthetic prokaryotic cell. Indeed, over the course of evolution, the host cell and its endosymbiont merged into a single organism. So when one prokaryote entered into another prokaryote, they established beneficial relationship in which both of them started depending upon each other, and with the passage of time this relationship became so much established and so much strengthened that eventually the two organisms became one, and that was the evolution of a eukaryotic cell. Now this figure explains the events which might have occurred in the ancient past. So this was the ancestor of eukaryotic cells, and this itself was a prokaryote, but it already had nucleus, endoplasmic reticulum, and nuclear envelope. Then this ancestor of eukaryotic cells, it engulfed oxygen using non-photosynthetic prokaryote, which over time became mitochondria, and so this was the beginning of the eukaryotic cell of animals, and then some of these cells engulfed photosynthetic prokaryotes. So this was the beginning of the eukaryotic cells of the plant. Now, first, the ancestral cell of eukaryotes, or the ancestor of eukaryotes, it engulfed an oxygen using non-photosynthetic prokaryote, which later became mitochondria, and then this cell with mitochondria engulfed another prokaryote which was photosynthetic in nature. So this was the beginning of the animal cell and the plant cells. So this theory is widely accepted theory till date. It gives the best explanation about the evolution of a eukaryotic cell from prokaryote ancestors, and this theory is consistent with many structural features of mitochondria and chloroplast. First, rather than being bound by a single membrane, like organelles of the endomembrane system, in the endomembrane system, ribosomes has no membranes, endoplasmic reticulum and Golgi bodies, vacuoles, lysosomes, they have a single membrane, whereas two structures, that is, nucleus has a double membrane, and here we are talking about the evolution of mitochondria and chloroplast. So these two structures also have double membrane. They are enwrapped, these structures are enwrapped in two lipid bilayer membranes. Now, there is evidence that the ancestral engulfed prokaryotes had two outer membranes. So, the ancestral cell of the eukaryotes itself was a double membrane structure. The starting cell, which engulfed the other cell. So the starting cell itself was double membraned, and as the mitochondria entered into that double membraned cell, it pinched off some of the membrane from the cell membrane of that cell, and therefore it also became double membraned. And similarly, when chloroplast later on entered into that cell, chloroplast also pinched off some of the membranes from that double membrane structure, and it also became a double membraned entity. So let me explain it with the help of drawing. So suppose this was the ancestral cell and it was a double membrane, ancestral eukaryotic cell. Now here the first structure, the oxygen using non-photosynthetic prokaryote, which later on became mitochondria. So as this mitochondria enters here, some of the membrane from this portion will be enwrapped around it. it just vanishes everything. So like this. So as this mitochondria entered here, this double membrane enwrapped around it, and similarly when chloroplast entered here, this double membrane also enwrapped around it. Therefore, the double membrane of these two structures validates the endosymbiotic theory. Now, what is the second evidence? The second evidence that mitochondria and chloroplasts were once upon a time free living prokaryotes is that mitochondria and chloroplast, they have their own genetic material, and the genetic material of bacteria, which is a prokaryote, is circular, single circular chromosome. So their genetic material is also in this form, and they have ribosomes, which gives an indication to the fact that these organisms were supplied with, these simple prokaryotes were supplied with not only their genetic material but also with ribosomes. So this is yet another proof that the which validates endosymbiotic theory. Now, the DNA in these organelles program the synthesis of some organelle proteins on ribosomes that have been synthesized and assembled there as well. So, mitochondria is also known as semi-autonomous organelle because some of the enzymes or the proteins which are vital for the survival of mitochondria and for its proper functioning are encoded by the mitochondrial DNA or the mtDNA, whereas it relies on the nuclear DNA for some of the proteins. A third and of probably this was the third which I explained. So they are autonomous organelles because they have their own genetic material, and they can reproduce through binary fission. Remember, bacteria also reproduce through binary fission. There is evidence that the ancestral engulfed prokaryotes had two outer membranes which became the double membrane of mitochondria and chloroplast. So it's the repetition of the first sentence, and I've already explained it with a diagram. Now we will focus on the structure of mitochondria predominantly, that how it looks like and how it performs the function it is made for. So mitochondria are found in nearly all eukaryotic cells, including those of plants, animals, fungi, and most protists. Of course, they are the powerhouse of the cells, and the cells do need energy for survival. So all the eukaryotic cells have mitochondria, and some cells have single large mitochondrion, but more often a cell has hundreds or even thousands of mitochondria depending upon the metabolic needs and the activity performed by a cell. The number of mitochondria also varies. For example, the cells that move or contract have proportionally more mitochondria per volume than less active cells. So metabolically active cells have more mitochondria. Each of the two membranes enclosing the mitochondria is a phospholipid bilayer, just like the plasma membrane or other membranes. But remember, once again I am repeating here, that the composition of the membranes surrounding various organelles is different, and unique set of proteins are embedded there and other structures are there. Why? So that this unique composition of the membrane also helps the organelle to perform its function properly. The outer membrane is smoother, but the inner membrane is convoluted and has several enfoldings which are called cristae, and these enfoldings are to increase the surface area for different reactions. The inner membrane has 4 complexes, and these complexes houses the various electron carriers. So these enfoldings increases the surface area so that the aerobic cellular respiration occurs at various locations. Now another point to note here is that the mitochondria can fuse with one another, and then they can separate from one another. So this is a kind of a dynamic process where individual mitochondria fuse together and then they separate from each other. The inner membrane divides the mitochondria into 2 internal compartments. The first is the intermembrane space. The space between the outer membrane and the inner membrane is called the intermembrane space, where the bulk of protons aggregate. And then the second compartment is mitochondrial matrix, that is the space enclosed by the inner membrane. The matrix contains many different enzymes as well as the mitochondrial DNA and ribosomes. Enzymes in the matrix catalyze some of the steps of the cellular respiration. Other proteins that function in respiration, including the enzymes that makes ATP, that is the ATP synthase, are built into the inner membrane. Mitochondria are generally in the range of 1 to 10 micrometer. If you remember, this is also the size of the bacterial cells. So this is yet again a proof that once upon a time mitochondria were free-living, simple prokaryotes. Now here they are referring to the inner membrane, which is thrown into several enfoldings to increase the surface area for cellular respiration, and this is yet another example of structure fitting the function. Now time-lapse films of living cells reveal mitochondria moving around, changing their shapes and fusing or dividing into separate fragments, unlike the static structures seen in most diagrams and electron micrographs. So these mitochondria are structures that are always in a flux. They are always undergoing continuous change. Sometime two mitochondria fuse with one another and became one. Sometimes many mitochondria fuse with one another and forms a net-like structure. So there is a continuous transformation in the shape of mitochondria. These studies helped cell biologists understand that mitochondria in a living cell from a branched tubular network that is in a dynamic state of flux. So, a flux, as explained, means something which continuously changes its shape. So, mitochondria is also a structure which continuously changes its shape. In skeletal muscles, this network has been referred to by researchers as a power grid. So, power grid is an interconnected network for delivery of energy, or if you consider electrical grid, then the delivery of electricity from producers to consumers. So once again, to refresh your memory, this is the structure of mitochondria. A single mitochondrion is shown here in the first image, and it has two membranes. The outer is smooth, the inner is thrown into several enfoldings, and these enfoldings are called cristae, and the inner space which surrounds the inner membrane is called the matrix, where the DNA and ribosomes reside, and there you have this dynamic change in the shape and the join fusion and then separation of the individual mitochondrial cells. So now we are done with mitochondria, and let's discuss peroxisomes. Peroxisome is a specialized metabolic compartment bound by a single membrane. Peroxisomes contain enzymes that remove hydrogen atoms from various substrates and transfer them to oxygen, producing hydrogen peroxide as a byproduct from which the organelle derives its name. So why peroxisomes are named so? Because they contain enzymes which remove hydrogen atoms from substrates, and then these hydrogen atoms are transferred to oxygen to form hydrogen peroxide. And these reactions have many functions. Some peroxisomes use oxygen to break fatty acids down into smaller molecules that are transported to mitochondria and used as fuel for cellular respiration. Remember that the beta oxidation of fatty acids gives acetyl coenzyme A, which is the which enters into Krebs cycle. So peroxisomes break fatty acids into smaller molecules and then deliver them to mitochondria, where they are incorporated into Krebs cycle through their derivative acetyl coenzyme A. Peroxisomes in the liver detoxify alcohol and other harmful compounds by transferring hydrogen from the poisonous compounds to oxygen. Now, the hydrogen peroxide itself is a toxic substance, but it is comparatively less toxic than the original form from which it was formed. Now, the organelle also is supplied with enzymes which can further uh, minimize the harmful effects of hydrogen peroxide by converting it into water. So, this is yet another example how the cell's compartmental structure is crucial to its functions. So inside the cell there are different organelles which are surrounded by membranes, and these membranes helps maintain an internal environment feasible for the organelle to carry out its functions irrespective of what is happening in the cytoplasm. The enzyme that produces hydrogen peroxide and those that dispose of this toxic compound are sequestered away from other cellular components that could be damaged. So, specialized peroxisomes called glyoxysomes are found in the fat storing tissues of plant seeds. So, with that, we come to the end of this lecture. For further details, you can refer to this book, and if you have any queries, you can ask through KCMS or WhatsApp groups. Thank you.